Hi, everybody. Welcome back. We are going to do the third part of the muscular system today. To kind of review what we have discussed in the previous section, I just want to cover the first few slides, or the last few slides we discussed. So we have talked about the connection between skeletal muscle and the neurons, which is part of your nervous system. We have talked about how electrical impulse or the action potential travels down your axon of the neuron, which reaches the axon terminal. At that point, you have the calcium voltage-gated channels opening up, causing the calcium to influx into your axon terminal. And upon that, you have the process of exocytosis of the vesicles that carry the specific neurotransmitter called ACH. ACH has the capacity to bind to specific channels located on the sarcolemma of the skeletal muscle. These channels you see right here are called sodium-potassium ligand-gated channel, or just simply sodium-potassium channels. And again, when the ACH binds to these channels, it allows both of the ions to move. So sodium is going to diffuse in, so you will have an influx of sodium at a higher level compared to potassium efflux, or moving out of the cell at a lower rate. The fluctuation in the charges causes the partial depolarization within the sarcolemma of the skeletal muscle. And that wave of depolarization can cause the sodium voltage-gated channels to open. Now you have a significant amount of positive ions rushing into your cell, which causes the depolarization phase of your action potential. To return things back to normal, you're using the potassium voltage-gated channels you see right here that allow the potassium effluxing and returning the ions back to returning the electrical charges back to the starting point. So you have depolarization and you have repolarization. Depolarization depends on sodium and repolarization depends on potassium. It's very similar to what we have described in terms of action potential for neurons in our nervous system. Now, when we go farther, the electrical impulses can spread throughout the skeletal muscle cell or your muscle fiber using these channels or T-tubules you see right here. And T-tubules connects to the sarcoplasmic reticulum. And within the sarcoplasmic reticulum, you had the calcium VGCs that are going to begin sensitive to the change in voltage, causing the calcium to rush from the sarcoplasmic reticulum, these really tiny red dots that you see. These red dots go and interact with troponin, and troponin moves tropomyosin, and that allows the binding site of your actin to interact with the myosin heads. And through this interaction and the movement of the myosin head, you create muscle contraction. This is really important. I really want you guys to be comfortable with understanding the process step by step. So make sure you spend some time understanding this in detail. Now let's assume that calcium is present in our environment. The myosin binding site of your actins are visible, as you can see in this picture. But understand that anytime I want to create muscle contraction, I require ATP. So how does that ATP get used? So we're gonna look at this process, which is basically forming a cross bridge and then creating something called a power stroke or a working stroke. So let's start here. When ATP is added, ATP causes the head of the myosin, or your myosin head, to separate from your actin. And this separation basically puts your myosin head on kind of a relaxed position. Notice how it's kind of sitting down close to the myosin itself. Now, as ATP starts to hydrolyze, which basically means it's going to break down, it will become ADP, again, losing one of the phosphates, right? So it becomes an inorganic phosphate and ADP, which is basically stands for adenosine diphosphate versus adenosine triphosphate. And this hydrolysis process of the ATP to ADP causes the myosin head to become high energy and go into what we identify as a cocked position. So again, you go kind of from this relaxed position to this cocked position where the myosin head is facing up. Now, because of the changing in the position of the myosin head, now myosin head can interact with the binding site on the actin, as you can see right here. And the interaction that is created is referred to as a cross bridge. Now, what causes the motion is as ADP and the inorganic phosphate leaves your myosin head, the myosin head creates its motion, and that motion again is called the power stroke, which literally grabs the actin and pulls it into the myosin, causing the muscle to shorten. Now, understand this, that when we're looking at this picture, by the end of this process, myosin and actin are interacting with each other, right? So if you want to continue pulling actin into the myosin, you need to allow those two to separate from each other first, and then you can grab a different part of the actin and continue pulling it in. Kind of think about it as a tug of war. So if you're pulling on a rope, right, and you're successfully pulling the rope towards yourself, you don't hold on to the same place on the rope. So you grab the rope, you pull your hand towards yourself, right? And then you let go of the rope and you grab another piece of rope and you keep pulling it. So think about the rope that you're holding as the actin, and your hands holding the rope as your myosin heads. So for actin to get pulled into the myosin, you need to create your pulling motion, which is your power stroke, then let go, and then grab another piece of actin again, bring it in, again, concept of tug of war. You bring it in, you let go, you grab a different piece, and then you bring that in. Now, how does that separation again happen? It happens by adding the ATP. So as ATP is added into your myosin head, again, that myosin and actin are separated from each other, and now you can start the whole process over again. Now, what triggers muscle relaxation? So typically we say muscle relaxation happens because the ACH, which is the neurotransmitter that basically started this whole process, is removed. Now, it could be removed by an enzyme called acetylcholinesterase, which is shorthand to ACH here. It could be diffused out of the site of the synaptic cleft where the ACH was released originally, or it can literally be uptake by the neuron to be used again later on. So it can be different means of ACH being removed, but again, the removal of ACH is what starts the whole process or stops the muscle contraction. So that means that when the electrical impulse does not reach the muscle kind of understands that the contraction is done. The calcium is collected back into your sarcoplasmic reticulum using an active process. If there is no calcium, there is no interaction with actin and myosin. And remember that the muscle is elastic, which means that if you basically don't have any connection between actin and myosin, your muscle recoils back to its original position. Now, when you think about your muscle contraction, one point that you do need to understand is when you look at the ratio between the skeletal muscle and the neurons that are innervating that, it cannot be one-to-one. So you basically, if you think about one single muscle or whole muscle in your body, let's say your biceps brachii, that is literally made up of thousands upon thousands of muscle cells. So you cannot provide one neuron for each of those single muscle cells. So what you do is you create something called a motor unit, and you can see it in the picture. So motor unit is one neuron coming out, but it has multiple axon terminals, right? And each of those axon terminals are now innervating a specific skeletal muscle. So basically, in another sense, one single neuron can innervate quite a few muscle cells at a given time. Now, if you need to create more force within your muscle, you literally recruit more of these motor units. So take a look at the picture I have down here again. Let's say you're lifting something relatively light. So the contractile force that you need only requires you to activate motor unit 1. But let's say you're adding weight on top of that, then you need to create a stronger contractile force. And in such scenario, you may activate your motor unit 2. Now, again, remember this, we talked about this in terms of nervous system previously. The motor units you may have in your face, which are going to give you basically your micro expression or a very small muscle movement. Those are going to be a smaller motor units. But when you're thinking about, let's say, your biceps, brachii, or when your quadriceps, right, your hamstrings, these are large muscles. So the motor units are much larger, as in one neuron supplying quite a few of those muscle fibers. Now, when we're looking at the action of the muscle, you could have something called simply a muscle twitch. So muscle twitch is when you have one action potential coming from your neuron, and that causes the muscle to contract and follow that contraction with a relaxation. This twitch can be absorbed in a machine that basically records those electrical impulses called myogram. And basically, again, is tracking the contractile ability of the muscle. Now, a muscle twitch typically has 3 phases: latent period, period of contraction, and period of relaxation. So latent period basically means it's delayed, right? So this is where the electrical impulse is basically passing through your muscle. That whole neuromuscular junction process taking place. So that's the excitation part. That's the depolarization phase. And that's going to be coupled with the contraction force that is coming up in the next phase. At this point, you really are not causing any muscle contraction. Your goal is basically to release calcium into your muscle that will lead to muscle contraction. Now, period of muscle contraction is where you actually have calcium, you have your cross bridges, you have access to ATP, and that causes the muscle to be able to create the power strokes and basically cross bridges that cause muscle contraction. And that's where you actually see the contractile force registered. And again, logically, after contraction, when the muscle starts to relax, you see the tension starts to decrease all the way back to 0. At this point, you're basically collecting your calcium back into your sarcoplasmic reticulum and basically stopping or halting the muscle contraction. So here again, you have the latent period, which is basically where the electrical impulses are taking place. You have the contractile force right here, or contraction phase, and then relaxation phase. And you can see on the x-axis, you have the time in milliseconds. So you can see the relaxation takes longer than the contraction and the latent period. Now, what if you have multiple stimuli in your muscle? So the red arrows you see down here represents the muscle contraction. Sorry about that. Describe muscle contraction or the stimulus that causes muscle contraction. So here again, you have kind of your muscle relaxed down here. So a stimulus comes in, right? You have your latent period, and then the muscle starts to contract, and then follow that with relaxation. Now notice as the muscle starting to relax, it never goes back all the way to the beginning phase or the zero tension phase, right? Before it reaches that, in this scenario, a new stimulus comes in, right? So your muscle is partially relaxed right before a new stimulus comes in. So in that scenario, then you will have another muscle contraction happens that is building up on the previous one. Because again, you didn't relax this fully, so whatever basically is left over of this, it adds up to the next muscle contraction. Again, same, muscle starts to relax, a new stimulus comes in. You take whatever you have here and you sum it up to the new one. So this process that we're describing here is known as lowest stimulation frequency, and it causes what we know as unfused or incomplete tetanus. That basically means the muscle never relaxes completely in this scenario, and the tension basically builds over and over again. So again, you never reach the maximum capacity of the muscle. The muscle still has the chance to relax, therefore it's called unfused or incomplete tetanus. But what is the opposite scenario? So what if you have a very rapid, not rapid, very rapid muscle contraction? So in that scenario, you basically will not get a chance to relax the muscle at all. So what you will have is all of the contractile force literally add up one on top of another. So look at the red arrows, see how close they are to each other, right? So you have basically the relaxed muscle here, your stimulus comes in, the latent period passes, and then you start to have muscle contraction. Notice there is no relaxation because again, the stimuli are so close together that it never gives the muscle to actually relax. In this scenario, you reach what we identify as fused or complete tetanus. So again, this is where your muscle is basically have reached its maximum capacity. Sorry, not maximum capacity, I should say that. It doesn't get the chance to relax because the stimulus are so rapid. Now, what happens is when you have so many stimuli coming in after another, what you reach what we identify as sustained contraction plateau, which basically means you have reached the maximum capacity. So as you can see in this picture, regardless of how many more stimuli come in, I have reached my plateau. Do keep in mind, guys, that when you do such a scenario, that these, if you allow the muscle to have continuous fused tetanus, that muscle is going to fatigue. So there is not enough basically energy or rapid supply of oxygen and ATP for the muscles to be able to maintain this contractile force. This is not something new in terms of how your muscle react to changes in terms of your activity, right? The stimulus strength. This is something you probably have noticed if you look around and basically look around to see if when you're lifting up something, right? So what we have is we have a concept called recruitment works on size principle. So if you are looking at lifting up something, your body says, well, I'm going to be conservative and I'm going to start with the smallest units of muscle that will be able to carry and complete the job by creating its contractile force. So it's going to recruit the smallest muscle fibers first. Now, as the job or the stimulus intensity increases, then there is more motor units that are going to be recruited that are going to be larger and larger. But you don't say, basically think about it, if you want to lift your cell phone from the table, you don't apply all the contractile force your biceps create, right? You just create enough for you to have the capacity to lift your phone. And a very interesting fact that the muscles of the arm, or any muscles in your body for that matter, do not contract all at the same time. They usually are not in, they're not, I guess they're not synchronous. They're asynchronous, which basically means you can have that muscle last longer because some muscle fibers are working and the other ones are resting. And then they kind of switch the job, right? So the ones that were resting come into work, and the ones that were basically tired, they will get rest. Now you can do it up to a certain point, but eventually if the stimulus is strong enough, that will lead to muscle fatigue. So it's not a solution that allows you to basically keep going and going and going. It's a way for you to prevent fatigue, or basically slow down the fatigue. Here's the same concept we were talking about in terms of size principle and recruitment. For a smaller job, you always start with the smaller motor unit, and if you need more and more, then the larger and larger motor units come into play. And the larger your motor units are, the stronger the tension. As you can see, the more tension you can build. Now, where do we get the energy to contract muscles? So we know that your muscles need ATP, right? Well, where does that ATP actually come from? So there are multiple ways, and there's three main ways that are described here. But even before we tap into the first one, you need to understand that your muscle always have some ATP stored in it. So that's a given. So if you look at this picture for about 6 seconds, as you start to utilize your muscles, you actually utilizing the ATP that is stored directly inside the muscle. Now, what happens right after that? Then you start to use a process called direct phosphorylation of ADP. And you're using a protein called creatine phosphate, or CP. And what that does is basically it takes the ADP and it adds an inorganic phosphate to it and makes ATP. And now this newly synthesized ATP can be used for muscle contraction. Now your body cannot rely on that for a long period of time. So then it starts to use a process called an anaerobic pathway. If you have ever taken typically a microbiology class or a lower division biology, the concept of aerobic and anaerobic should sound familiar to you. Anaerobic implies that there is no oxygen involved, and the way that our body uses anaerobic pathway to create energy is a process called glycolysis. Now, we'll talk about glycolysis in detail when we talk about metabolism later on. But right now, what I want you guys to understand is if your body is doing an anaerobic process, that is the process of glycolysis, and it does create lactic acid throughout the process as it is producing that extra ATP. Now, these three mechanisms, basically the stored ATP, creatine phosphate, and anaerobic pathway, basically give your body and your muscle enough time to activate the most effective way of producing ATP, which is aerobic respiration. Aerobic respiration implies that your body is actively using oxygen, and through usage of that oxygen, it produces a significant amount of ATP. And again, we'll talk about that in the metabolism lecture. So look at this picture. Again, you have ATP as the beginning, and then creatine phosphate right after. Then you start to use anaerobic pathway. And sometimes, if your activity is relatively fast, you don't even kick into your aerobic pathway. You may finish it. But if you do have prolonged level or prolonged activity, then you are actively tapping into your aerobic process and using oxygen to produce significant amount of ATP. So kind of looking at this picture, let's say you're running 100 meter, right? Versus you're doing a marathon or a 40-minute jog or whatever, right? So that's the main difference here. So if your activity is fast, you don't even tag into an aerobic process. Now, kind of building on that whole concept of aerobic and anaerobic, that is one way for us to actually classify our muscle fibers. So our muscle fibers, there are actually 3 different types, and this classification comes from how they're manufacturing their ATP mainly, and how quickly they are contracting. Again, contractile force, think about it as, are they contracting fast and can basically do an action or create a significant amount of contractile force over a short period of time? Or are they going to contract slowly and provide contractile force over a longer period of time? So let's just start by how they manufacture energy, or your ATP. Again, if they are using oxygen, which is an aerobic process, these fibers are called oxidative fibers. If they're relying on glycolysis or an anaerobic process, they're called glycolytic fibers. So before, again, I'll move on to discussing that with you, let's make sure we have an understanding that if you are using oxygen, you have to be able to store that oxygen inside your cell, which means we will have myoglobin at a higher concentration. Because we're using oxygen and oxygen gets transported by your blood vessels, we will have a higher amount of circulation within these muscles, so a lot more capillaries. And the amount of ATP that can be produced during an aerobic process is a much higher level compared to if you're using an anaerobic process. So let's take a look at these 3 categories of muscles: a slow oxidative versus fast oxidative versus fast glycolytic. So look at the name, guys. Let's just start with the first part, a slow and fast. So a slow, again, it's going to contract slowly, or the contractile force is less, versus fast glycolytic is going to contract rapidly with a relatively large contractile force. Oxidative implies that it needs oxygen, versus glycolytic, it means it's using glycolysis. So a slow oxidative, how are you characterizing them? Again, red color due to abundance of myoglobin as they store oxygen. As the name also implies, they are receiving they're producing their ATP through an aerobic process. They're going to have larger number of mitochondria because mitochondria is the organelle that uses the oxygen to produce ATP. Because they have high levels or high demand for oxygen, they will have more capillaries. And the word slow, again, implies that they contract slowly, but because they have easy access to energy, they can last for a longer period of time. The fibers on these muscles are relatively small in diameter. Fast glycolytic is almost opposite of what we talked about, the slow oxidative. So they contain little myoglobin and a few mitochondria. The diameter is larger compared to slow oxidative ones we were talking about. They do contain more myofilaments, which are basically actin and myosin, which gives the muscle to create a stronger contractile force or more power. The word glycolytic in the title implies that they are using anaerobic process. And again, because glycolytic does not produce as much ATP, and this type of muscle burn through a lot of ATP fast, these muscles have the tendency to get tired fast. Last category of muscles we have is fast oxidative fibers. These are intermediate diameter, so they're kind of between the slow oxidative and fast glycolytic muscles. As the name fast implies, they do contract rapidly. However, because they do have access to oxygen, because of oxidative process, they don't get tired as fast. They are oxygen dependent. They have high levels of myoglobin because they're oxidative, right? And they also have rich supply of capillaries to get that oxygen in the first place. Again, because of the high access or easy access to high levels of ATP, they are relatively fatigue resistant, even though they are fast. And they are identified as the more powerful type of muscle when compared to slow oxidative fibers. Okay, a couple last concepts in terms of muscle identification and where the actions, origin, and insertion words come from. So every time you think about a muscle, you need to remember that the muscle has multiple points of attachment, and these attachments can be either identified as origin or insertion. Now, origin implies that the point of attachment is less movable or immovable. Or when we're talking about insertion, that means that that attachment during the muscle contraction is more movable. So take a look at the example of the muscle called brachialis. So brachialis as one end connects to your humerus, and on the other end connects to your ulna. So notice that, first of all, the type of connection is also different. So this is identified as direct versus an indirect attachment. And I'll talk about that in just a minute. But if you move or contract this muscle, your ulna gets closer to your humerus, right? So here's basically what we're talking about. However, the position of your humerus doesn't change. That means the humerus position or attachment will be identified as origin, which is less movable or immovable, versus ulnar attachment, which is moving, is identified as your insertion. Now, as I mentioned earlier, we have something called direct versus indirect attachment for the muscles. So looking at this, again, this is a direct attachment, this is an indirect, and you can clearly see a difference between the two. So what are these? So direct or fleshy attachment is when muscle surface is attached directly to the periosteum of the bone. So if you guys don't recall periosteum is the connective tissue that is wrapped around the bone. Now, if this direct attachment or this fleshy attachment is absent, we identify that as an indirect attachment. The indirect attachment is usually identified as a tendon or aponeurosis. So tendon is typically long, cord-like fibrous structure. It is made up of regular connective tissue. And aponeurosis is usually a very wide, thin tendon. So the best example of the aponeurosis is the one that covered the skull. And again, this is a really good example that holds basically the frontal belly and the occipital belly of the muscle that covers the surface of the cranium. Now, if you guys kind of think back at your skeletal system, we learned some vocabulary like tubercles, trochanters, crest. Basically, if you guys recall, these are going to be points of attachment where the muscles can either directly or indirectly attach to the bone. Now, how do I name a muscle? There are actually different qualities or characteristics you can use to name a muscle. For instance, you can use its location. So when you say a muscle name is tibialis anterior, basically means that this muscle is located in front of the tibia. You can use the size of the muscle. So you can use such words such as maximus, minimus, medius, longus, brevis, major, minor. You can point out the direction the fibers are moving. If they are going straight down, parallel to each other, we use the word rectus. If the pattern is horizontal, we refer to this as transverse. And if the pattern is more in angle or sideways, they refer to as oblique. Number of origins. So you have muscles called biceps or triceps. That basically means how many heads or points of attachment they have in terms of origins. And then shape of the muscle. Muscle called deltoid is triangular, and the word deltoid literally means triangle. And serratus means a sawtooth pattern or seesaw pattern. These are muscles that are on the side, and you can again see that serrated pattern on the muscle, hence the name serratus. Now there is these two pictures describe some of the most important muscles we have. Most of the muscles that are listed here are superficial muscles. However, and this is a really good picture both from the anterior view and the posterior view to review the muscle names that you guys need to know for your lab. So use this as a good resource in terms of muscle identification. And that's about it for our muscular system.